All right. So what we're going to do today is continue wrapping up on the human microbiome, and then talk a little bit about symbioses, which I introduced already, but I'm just going to quickly reintroduce it before we spend some time talking about negative associations between microbes and humans. So continuing on the microbiome, I know you really wanted me to go into the do-it-yourself fecal transplants again. That's where we left off, but thankfully I will not talk about that anymore. So in the previous lecture I talked about the human microbiome, and I tried to emphasize taking an ecological perspective on thinking about the human microbiome. Because it's an ecosystem, that ecological perspective can help greatly with our ability to study and ability to design experiments and our ability to understand what's going on in this cloud of microbes that live in and on people. And what I want to talk about for a few minutes here is also the importance of thinking about evolution in the context of the human microbiome. And everything I'm talking about here would apply to communities of microbes that live in and on other organisms. We're not going to talk about that in detail right now, but there's a cloud of microbes that are on every type of animal and every type of plant, and even many of the single-celled microbial eukaryotes are surrounded by a cloud of microbes that the general approach that we're doing here applies to all of those other systems. So in the context of evolution, one thing to think about is that you can treat aspects of the microbial community as traits and certain components of the microbial community as character states, and try and look at the evolutionary history of the microbial community by characterizing microbial communities in different organisms and treating some aspect of the microbial community as your input for a data matrix, and then trying to infer what the history of the microbial community was. And, you know, we can do that for humans in the context of knowing something about human, chimp, gorilla, orangutan evolution. And you should be aware that historical studies and evolutionary studies of ecosystems can be very important for understanding the development of those ecosystems as well as the interactions among organisms. And we'll talk a little bit about this in the next few lectures. I just thought I'd show you some actual data from some real papers on this topic. This is a paper by a scientist named Ruth Ley and a bunch of colleagues, where what they did basically on the x-axis are what samples they were looking at, and that included water samples, soil samples, various parts of the human body like skin, salt water, termite gut, etc., and then gut samples, well actually fecal samples that are thought to tell you something about the gut samples from across vertebrates. And the colors correspond to different phyla, which exact one doesn't really matter here. And what you see is lots of variation in microbial communities when you sample them from all sorts of different environments. But when you get into the vertebrate gut, you see the vertebrate gut across almost all vertebrates that have been looked at is dominated by a couple of phyla, firmicutes in blue and Bacteroidetes in red, and then smaller amounts of a few other phyla, and then not much else. So the vertebrate gut ecosystem is a very narrow representation of the total potential diversity that is out there in the world of bacteria. And this tells us, or at least provides us hypotheses about how we and other animals regulate what microbes sort of show up and live in our community. It's not that any microbe that shows up in our gastrointestinal system can take hold there and survive there and thrive there. This is a very specific niche that has been there for hundreds and hundreds of millions of years, and this community has been co-evolving with vertebrates for a long period of time. And I just thought I would bring in some recent news. There was a story going around the web in the last couple of days about a new scientific paper where people were trying to zoom in on this branch, zoom in on the human branch, and try and sample people who have not been exposed to many of the sort of modern trappings of civilization. In particular, what people are really looking for is people that have low exposure to antibiotics and low exposure to certain aspects of the diet of Western culture. And so there have been a bunch of studies in the last few years where people have been trying to go to relatively isolated indigenous cultures in a variety of places and try and look at the microbial community in these people and compare it to the microbial community in more developed areas. And that's what this recent study was. It was of a hunter-gatherer population from Tanzania, the Hadza hunter-gatherers. And this paper just literally came out a couple of days ago. And there have been some really interesting news stories about the implications of this study. And what they basically did was go to this community in Tanzania and work with the people there actually to get information about their diet, information about various behavioral practices, as well as collect samples for microbial studies. And what this plot shows, just like some of the other plots, is the colors represent relative abundance of different microbial phyla or major lineages of bacteria and archaea. Again, if you remember, the way you determine relative abundance is you take a sample, you isolate DNA from that sample, you run ribosomal RNA PCR, you read sequences, you build an evolutionary tree, and you map each individual sequence to a phylum. And then you count how many of your different sequences show up in each of those phyla. And that gives you the number that you show here. The height of this bar corresponds to the number of counts that you get for these different samples. And over here on the right is a collection of samples that they took and applied the exact same protocols to, the same methods to collect the samples, the same methods to isolate the DNA, the same methods to run the PCR reaction. This is really important. Different labs do things in different ways, so it's important to try and have a control for your study. So this is a collection of Italians. This is a collection of the Hadza hunter-gatherers. And certainly you can see some similarities between these populations, but the Hadza have a much greater diversity of microbes in their gut samples and a much higher relative abundance of some particular taxa, like spirochetes, shown here in red. Proteobacteria, shown here in yellow in particular, are much more abundant, as opposed to in the Italians, the same sort of proportions are taken up by this high GC gram-positive group. So there's some serious differences in the microbial communities between sort of modernized populations and people who have not more isolated indigenous populations. This is the same information, same study, but then replotted rather than by phyla, but by genera. So trying to assign individual sequences with the same approach into particular genera of microbes like Escherichia or Bacillus, etc. And again, you see pretty big differences between the Hadza and the Italian populations. Now, this doesn't say what they're functionally doing in these communities. It doesn't even say how the development has happened, but we need to know sort of the information about different communities in order to then think of experiments to do that would then test what the functions are of these communities. And there are probably in the next year or two going to be hundreds of studies like this that are coming out. There are people working on mummy samples. There are people working on human samples that came from frozen ice environments, that people from ancient history samples, as well as samples from isolated populations. And we're going to learn a lot more about the human microbiome via this. This is just showing the same data in a different way. This basically shows that the Hadza are really different than the Italians, than people from the United States, as well as from other indigenous populations. And one of the reasons that people are really interested in doing this is to try and use some type of character state reconstruction method to figure out what the ancestral microbiome was like in humans in order to try to understand whether or not some of these ailments that I was talking about in the last class, like allergy, autoimmune diseases, obesity, and other phenotypes that are increasing in their frequency in the human population, if we can then compare the individuals with those diseases or phenotypes to each other and try and identify if they have altered microbial communities, and then ask, is that alteration something like they've lost certain microbes that were present in the common ancestor of humans, or is it some new gain of new microbes that might be causing some negative effects as opposed to losing some microbes that might have been causing positive effects? So are there any questions about that before we move to talking just a little bit briefly about functions of microbial communities in humans? So I'm not going to show you the data behind all of these different studies, and I don't expect you to know all of these different effects of the microbiome. I just thought it would be interesting to people. So in the last 20, 30, 40 years, there have been a variety of studies that have started to accumulate information about the potential or known functions of the human microbial community that is, again, this sort of cloud of microbes that lives all over us. It's been well known for many years that the microbes in our gut help with digestion of food. They profoundly impact things like the metabolism of drugs because they're involved in converting compounds that are in drugs into other compounds. Some of the things I was mentioning related to autoimmune disorders are that the microbial community affects the development and regulation of the immune system. There's lots of strong evidence that certain aspects of the microbial community can help prevent infection by pathogens. There's some indirect evidence that the microbial community is involved in healing of certain types of wounds. There's certainly, as I showed you, that obese mouse, and there's lots of suggestions from humans that the microbial community influences metabolic rate, uptake of calories, and therefore might have a big impact on weight gain or weight loss in individuals, and so on. There's lots and lots of both established functions of the microbial community as well as suggested functions of the microbial community. And there's more and more information every day accumulating about this. So I think that the human microbiome is going to become much more important in discussions of human health and medicine, to the point that there are many people that are now making the argument, not sure how I feel exactly about this argument, that the microbiome could be viewed in essence much like an organ. It has incredibly diverse functions. Those functions and the community that we see in people is very tightly regulated developmentally in individuals. It's not a random collection of microbes, and that you might want to start thinking of the microbial community in different parts of the body more like an organ than like accidental visitors that are showing up just living in your system. I didn't really mention this before, but if you look at not just the number of organisms that are in our system, so you're covered in tens of thousands of species of microbes, but if you look at the gene content in those microbes, the total number of genes in the microbes that live in and on a person is much greater than the total number of genes in your genome. And there are incredibly diverse functions encoded by those genes. And so understanding human biology is going to require understanding what the functions are of all of these microbes, not just the functions encoded by our genomes. And I just thought in terms of function, I would just leave you with one other. The latest hot fad in microbiome studies is there's a bunch of lines of direct and indirect evidence that the microbial communities in the gut may be manipulating behavior in a variety of ways. So there's been a well-known sort of what's called the gut-brain axis for communication of nervous system signals and some hormonal communication. If you alter the microbial community in the gut, that definitely alters that signaling system, and there have been multiple studies that have now shown in animals primarily that behavior of an individual can be affected by the microbial community in their gut. this shouldn't be that surprising. We know that pathogens, that is, organisms that make us sick, affect behavior in many ways. Rabies is a classic example, but there are many others. So it shouldn't be that surprising that the microbial community might be manipulating the behavior of their host in a variety of ways. And then just before we get into the pathogen studies, I just wanted to point out that a lot of the studies of the human microbiome have been what are called correlative. That is, you collect information about a bunch of different people. I'll give you one classic example. There's a lot of studies in the last year showing that individuals with autism have a different microbial community than individuals who do not have autism. That's a correlation. That does not mean that alterations in the microbial community cause autism. The alteration in the microbial community could be just yet another symptom of the disease behind autism, rather than the actual cause of this. So it's really important when you look at things that are complex like this to not confuse this correlative study with information about causing particular phenotypes. And then if you're interested, I thought I would put this up here, and for full disclosure, I'm involved in both of these projects as an advisor, but you can now, I don't know how many people, has anybody here done the human genome testing, like via 23andMe or other? Yeah, so I did this, and you basically do a spit in a tube, and then you get data about your genome back from a variety of companies. There's one called 23andMe. These are new startup companies. There are actually about 10 of them now that will do the same thing for your microbial community, and will characterize the microbial community in a sample and send you back a report and sort of tell you a little bit about what microbes they found in your sample. Not clear if that has any use in medicine at this point, but it's kind of cool. All right, so we're going to switch now to talking about symbioses, and in particular to talking about the negative interactions between microbes and humans. So just a reminder, I think I've defined both of these before. Symbiosis, in the formal way we're going to use this in the course, is any type of intimate association between at least two organisms where at least one of them benefits. And I like to sort of put this up there because many people use the term symbiosis to refer to mutualistic interactions where both partners benefit. And we take a broader definition of symbiosis where it's only one of the two partners that has to benefit, and the other partner in the situation might benefit or might not benefit. And endosymbiosis is basically the same thing, but it's symbioses where one of the partners is inside the cells of another partner, like the organelle examples that we talked about extensively in previous lectures. So again, if you have one organism benefiting, the effect on the other organism allows us to classify symbioses into different categories. Mutualism is when both partners benefit, and there are a litany of examples, many of them in the book, some you will hear in the rest of the course, about different kinds of mutualistic interactions between bacteria, occasionally also archaea, and multicellular eukaryotes. There are lots of other types of mutualistic interactions, but these are sort of particularly striking and interesting, where there's some plant or animal or other multicellular eukaryote, and they acquire some function via symbiotic interactions. There are a lot of well-known digestive mutualistic interactions where, for example, in the gut of ruminants, that's how cellulose is digested. Animals can't digest cellulose themselves. They always have to have a microbial community that helps them do that. So termites, for example, wood-boring beetles, anything that eats grass. So all of the ruminants have cellulolytic bacteria that live inside of their gut or in some other part of their system. There are a good example in chapter 26 about this behavioral symbiosis where there are these bobtailed squid that use a bacteria, a Vibrio type of bacteria that is bioluminescent, and they use this bacteria to provide counter lighting when they're swimming around at night. so that they light up sort of their bottom of their body, and when they're swimming around, if there are organisms below them looking up towards the moon, they can't recognize that there are these squids swimming by because they have this lighting that compensates for the shadow that the animal would be developing. It's an incredibly cool symbiosis between a bacteria and these squid. There are lots of autotrophic symbioses where a bacteria usually, sometimes a photosynthetic eukaryote, is providing photosynthesis for a host. We talked about lots of these with the organelle evolution in chloroplasts. There are many others that are out there, and this example that I've mentioned a few times is really interesting. In the bottom of the ocean, there are these giant tube worms that have no mouth and no digestive system, and they basically function in a way like plants. They have, instead of photosynthetic bacteria inside of them, they have chemosynthetic bacteria that fix carbon and provide all the nutrients for their host, much like chloroplasts do for plants. And again, there's lots of these covered in chapter 26 and in other places in the book, and we'll hear more about them in the rest of the class. The human microbiome story I tried to sort of say is categorized frequently as commensalism. Technically, commensalism is really when one of the partners benefits and the other partner we don't really know does not benefit. For the human microbiome example, we don't actually know whether either of the partners benefits in some of the cases, and we certainly don't know a lot about the exact type of interaction, and we generally use the term commensalism, although that might not be quite accurate. And so we're going to spend the rest of the class today on is parasitism. So a symbiotic interaction, an intimate association between some organism where one of them benefits and one of them is negatively affected. In this case, the negatively affected one we're going to focus on is humans, and the one that benefits is going to be some type of microorganism in the partnership. There are many different ways that an organism can negatively affect a host in some way and positively benefit. We're going to focus on a particular sort of subclass of parasitism where the organisms are called pathogens, and the real difference here is that the pathogens have to be an infectious agent. That is, they can be transmitted between one host and another host. We'll come back to this in a minute. And they have to cause some type of disease in the host. So we're going to focus on this sort of subclass of parasites called pathogens. Now, we've already seen many examples of this. When I went through the list of different lineages of bacteria, I included on some of the slides examples of pathogens in those groups. So the spirochetes include the causative agents of syphilis and Lyme disease and many other human diseases. The chlamydias include the causative agents of many human and animal diseases. The high GC gram-positives include the causative agents of tuberculosis and leprosy, as a variety of others. The low GC gram-positives include many pathogens, anthrax, etc., proteobacteria, many pathogens. There are also many pathogens in the eukaryotic groups. So in the apicomplexans, basically all of them are pathogens. I didn't mention these. I've added this onto the slide. There are pathogens within the ciliates. So the ick, if you have fish tanks, there's this weird, gross thing called ick that grows on many of the tanks in the fish. It's actually a ciliate, pathogenic ciliate. The oomycetes are basically the best characterized ones, are a variety of pathogens. The diplomonads and parabasalids include many pathogens. The kinetoplastids include many pathogens, and so on. The heterolobosians. I mean, there are pathogens scattered throughout the tree of life. And if you look inside many of the bacterial groups, most of the species in those groups are not pathogens, but there are pathogens in many of the groups. In the eukaryotic lineages, there are some where all of the members are pathogens, but again, it's in most cases some pathogens, but many non-pathogens. Something very unusual that nobody yet has a good explanation for, there are no archaeal species that are known to be a pathogen of anything. no good explanation for this. If you figure this out, you will get great fame and probably win all sorts of awards of some kind. There's been lots of speculation about something about the membrane structure of archaea or something about the way they interact with other organisms, and I've yet to see any good information about that would explain this. So chapter 26 in the book has a description in more detail of this concept called Koch's Postulates. It's named after the person who developed this, and basically about 100 years ago, he came up with sort of a set of rules by which you could define an organism to be a pathogen. And the book walks you through what those lines of evidence that you need in order to officially determine that a particular organism is a pathogen and also a particular disease is caused by a pathogen. And what I'm going to do is give you a case study of this. It's also one they talk about in the book. It's a very interesting story. It involves gastric ulcers. So this is an ulcer in the stomach. An ulcer is basically a small hole, and a lot of people get these gastric ulcers. For many years, for hundreds of years actually, the cause of gastric ulcers was thought to be stress. And when someone got an ulcer, they recommended you reduce the stress in your life and do things to relax more. You know, it's always the person's fault in some way to get a disease. And in the 1950s and 1960s, a variety of researchers started to find interesting patterns where if you sampled sections of the stomach where there was a gastric ulcer, there were very weird spiral-shaped bacteria that were found in these sections, and it was unclear if they were a symptom or some people suggested they might be a cause of this disease. And so it turns out that these bacteria are not spirochetes. They were originally thought to be spirochetes. They're a proteobacterium that has a spiral shape like spirochetes. And people eventually cultured it, got it to grow in the laboratory. It got a name, Helicobacter pylori. And one, or actually two scientists in particular became a bit obsessed with this organism. It's a proteobacteria, just to remind you of that. So this one scientist, Barry Marshall, became obsessed with this and wanted to prove that it was the causative agent of stomach ulcers rather than a symptom of stomach ulcers. And so he experimented on himself. He grew up some Helicobacter pylori from a person with a stomach ulcer, and he drank it. And he got stomach ulcers. And then he cured himself of this with antibiotics. So this proved for the first time that these stomach ulcers were not caused by stress, but were caused by this bacterium. He won this shiny metal Nobel thing for doing this. Has become quite well known in the community. I mean, sounds a little deranged. It is a little deranged, but it is a remarkable story of, willingness to test to the extreme a hypothesis about the causative agent of this disease. The book walks you through a little bit of Koch's postulates in terms of Helicobacter pylori. I'm not going to go into this in detail here, but it outlines, the rules and how the rules were tested for Helicobacter pylori. The microorganism must be present in every case of the disease. It must be cultured from a sick host. It must be able to induce the disease. That's him drinking the Helicobacter pylori. It must be recoverable from newly infected individuals after you transfer from an infected individual, and so on. And this is how he proved it, and then won the Nobel Prize for this. So it's a pretty amazing story about a human pathogen. Just as an aside, before we go into talking a little bit about anthrax, Helicobacter pylori, there are, just like we talked about Escherichia coli and how two different strains of E. coli can have big genetic differences, same is true for Helicobacter pylori. There appear to be strains of Helicobacter pylori that do not cause stomach ulcers, that may in fact be mutualistic symbionts, and there are varieties of Helicobacter pylori that cause stomach ulcers and also can cause stomach cancer and are therefore pathogens. And they differ in regions of the genome that have been acquired by lateral gene transfer. So I'm going to do another case study here of a human pathogen. In this case, I would recommend against drinking anything containing anthrax in a sample. Anthrax is a member of the Firmicutes phylum. So we've talked about the Firmicutes before, the low GC gram-positives. Anthrax is caused by a bacterium called Bacillus anthracis. It sporulates like other members of the Firmicutes phylum. It has been used, unfortunately, as a biological weapon by many groups, or at least developed into a biological weapon by many groups, in particular because these spores are very resistant to heat. People thought you could put spores of anthrax inside a bomb and distribute it via a bomb, and therefore, cause anthrax in a population of people. I don't know, this is probably too long ago for many people here. I don't even want to know how old people were when this happened. But I was working at so I wore this shirt today. I used to work at a genome institute called TIGER, the Institute for Genomic Research. And when I was working there in about 1999, the institute got a grant to start to study the genome of Bacillus anthracis, because there's a lot of interest in protecting from biological weapons attacks, and also because it's actually an interesting and major pathogen. And then in October, just after September 11th, so just a month after September 11th, letters arrived at a variety of facilities, including the US Senate, that contained a white powder inside the letters. The white powder was spores of Bacillus anthracis, and I think 4 or 5 people died as a result of exposure to these spores from these letters. Created a major federal investigation, led to all sorts of changes in how researchers work with a variety of pathogens. And what happened, the institute that I was working at, because we were the only place in the world that was already working on looking at the whole genome of Bacillus anthracis, got recruited basically by the US government, by the FBI, and by other agencies to characterize the anthrax from these letters in more detail. And so what I'm going to do is tell you about the DNA-based characterization of anthrax in order to try and resolve where the anthrax came from in these letters. So the first question that was asked basically by everybody involved was, how do you figure out where this anthrax came from? And it turns out the answer came from phylogenetic analysis, from building evolutionary trees of the anthrax in the letters and comparing it to the anthrax from other samples. And I'm going to back up for a little bit. So while we were working on the genome of anthrax, there was one researcher in particular, Paul Keim, who's at Northern Arizona University, who had been gathering anthrax samples from all around the world. Anthrax can kill cattle and sheep. People have isolated anthrax from all sorts of places all around the world. And studying the diversity of anthrax in a variety of ways. And if you wanted to study the diversity of anthrax and build evolutionary trees of anthrax, one thing you might want to do is extract DNA, run PCR, do ribosomal RNA sequencing, build a data matrix, and then build an evolutionary tree of different strains of anthrax and see how they relate to each other on this tree of ribosomal RNA. And so that's what some people did. They took these isolates of anthrax, read the sequence of 2 different strains of anthrax and then 3 different strains of anthrax, and built evolutionary trees based upon the ribosomal RNA, and it turns out this didn't help. All different varieties of anthrax are identical in their sequence for the ribosomal RNA gene. The ribosomal RNA gene does not evolve very rapidly. It evolves enough so that we can build evolutionary trees and say that, this organism is a bacilli, or this organism is a firmicute, or this organism is a bacterium. But it doesn't evolve rapidly enough to distinguish 2 different strains of anthrax from each other. And so what Paul Keim did was go to different parts of the genome of Bacillus anthracis that evolve more rapidly than the ribosomal RNA genes. Ribosomal RNA is very constrained by selection because it functions in this very important process of translation of RNA into proteins. it just can't change very rapidly without killing the organism. But there are lots of parts of the genome of a bacterium that can change more rapidly. And so you don't need to worry about all the details on this slide, but what he did basically was find regions that changed rapidly. They're called variable number tandem repeats, or VNTRs. They change in the copy number of a short repeat sequence in the genome. So you can have GA, GA in one organism and GA, GA, GA in another organism. And you can count the number of copies of this particular repeat in different strains of anthrax, compare them to each other, and then build evolutionary trees based upon this. And he surveyed about 50 different regions of the genome that contained different versions of these repeats, and then counted the number of copies, built a big data matrix, and built big evolutionary trees of this. I didn't mention that anthrax is culturable, so you could grow the anthrax in the laboratory and then do the experiments sort of the easy way, not doing environmental surveys, but doing it based upon cultures of anthrax from different areas. And he made this really nice tree based upon these VNTRs that classified different varieties of anthrax into different parts, different monophyletic groups within the phylogeny of the Bacillus anthracis lineage. And so they're labeled here. The labels don't really matter. Some of these are different locations. Some of them are different flavors of anthrax in essence. But that's the equivalent of building a tree with ribosomal RNA, but now with finer resolution to understand the different varieties of anthrax and distinguish them from each other. And then he characterized the anthrax in the letters and mapped it to this one particular group within the anthrax lineage. Turns out this was, I guess, both good and bad when this was found out as part of the investigation. So when he mapped the anthrax in the letters to this particular clade, it turns out that particular clade includes the variety of anthrax that all labs in the United States and in other countries were using as a model strain to study the biology of anthrax. And it was also the same strain of anthrax used in many biological weapons projects. So what this told people was that whoever put the anthrax in the letters had access to this particular strain of anthrax, known as the AIM strain, used in labs throughout the world. It wasn't someone going out and collecting anthrax from a field and growing it up. They somehow got access to this laboratory strain of anthrax. People have characterized actually the written history of the distribution of this particular AIM strain. It came from a cow that died of anthrax and then was sent to a US research lab, Fort Detrick, and then distributed to other labs around the country and eventually to the United Kingdom and then eventually to other locations. And what the FBI then recruited this institute where I worked to do was to sequence in more detail. So I should say the VNTRs are identical between the different versions of the AIM strain. So here's the AIM strain. It was sent around to different labs, and every one of them had identical patterns for these VNTR loci. But these strains have been grown in different laboratories for many years. And it seemed likely that during that growth, they may have gotten a few mutations in other parts of the genome that could be used to identify where the anthrax in the letters came from by sharing mutations that happened during the transmission of these strains or growth in different laboratories. So the institute that I worked at then got a grant to read the entire genome sequence of the anthrax strains from the letters and from different samples of the AIM strain that came from different laboratories around the world. And eventually the people who worked on this, colleagues of mine, built an evolutionary tree based upon the complete genome of strains of the AIM strain. It was the only way to resolve where the Florida isolate was in one of the letters. And they mapped 3 particular samples from labs around the world, B, C, and D samples, that were most closely related to the anthrax in the letters. And this helped with the investigation to try and figure out who was behind the sending of the letters, the anthrax letters. Turns out there's still some controversy in this story of the who did the sending these anthrax-containing letters around, because some of these strains, many people had access even to these B, C, and D isolates. So this doesn't tell you who did this. It just tells you where to focus the research and the criminal investigation into. If anybody had access to this D isolate, which unfortunately was hundreds of people, they are now potentially the suspects for this investigation. So phylogenetic analysis was essential. It was used in Senate investigation into this. It was used by the FBI. It was used by court cases associated with this investigation. Are there any questions about this before we move on to the last sort of story about viruses? Don't drink anthrax. So we've talked a little bit briefly about viruses before, and I mentioned these 3 alternative theories to explain where viruses came from. Viruses are unusual, but they fit into this pathogen section of the course because all viruses are obligate parasites of other organisms, and some of them are infectious and cause disease, so they would fit in the category as pathogens. Where viruses came from is still a bit of a debate. There are multiple theories about this. I've mentioned this before. It's hard to resolve the exact evolutionary history of many viruses compared to cellular organisms because they don't have ribosomal RNA genes and they don't contain many of the universal homologies, the genes that are behind universal homologies, that if we look inside the sequence of those genes, we can build evolutionary trees of bacteria, archaea, and eukaryotes, but we can't include most viruses in those trees. But nevertheless, they're really interesting for evolutionary studies. There's lots of interesting stories about their evolution because they all have genomes, they all undergo mutation and diversification, and they all evolve. Again, I mentioned this before. I'm not going to go into a lot of detail. I'm just going to actually skip over these different models for viruses so we can talk about this one last story that I want to talk about. You can ignore these other Mimi virus things. I want to talk about this last story about the evolution of influenza viruses, where phylogenetic analysis has also been and continues to be critically important for understanding the flu virus. So the flu virus causes influenza. The term comes from basically meaning bad air. It's a particular type of virus called a negative strand RNA virus. The genome of influenza is made up of RNA molecules. The reason it's called negative strand is they have to be first copied back into the, quote, positive strand before they are used to make certain types of products. Influenza viruses have 8 RNA chromosomes, and those chromosomes encode the different proteins and RNAs that make up the biology of influenza. And there are 2 of them in particular, 2 of the proteins encoded by influenza that are very well characterized. One is called hemagglutinin, and one is called neuraminidase. They are abbreviated H and N. That's where the H1N1, for example, H5N1, etc., comes from for classifying influenza viruses. That's done by looking at the hemagglutinin genes and the neuraminidase genes and identifying which, in essence, monophyletic group of each of those genes that virus has for its hemagglutinins and neuraminases. And what researchers now do routinely is if there's a flu outbreak or even before there are major flu outbreaks, they screen for flu in a variety of populations. They sequence the chromosomes in particular that encode the hemagglutinin and the neuraminidase genes, build phylogenetic trees of those genes, and that allows you to understand what type of virus you are looking at. And what's somewhat unusual and also responsible for many of the problems that flu causes is that by doing this phylogenetic analysis, scientists have found that the phylogenetic tree of the hemagglutinin genes does not always match the phylogenetic tree of the neuraminidase genes. That is, inside the genome of flu, different genes have different evolutionary histories. It's very much like the organelle story that we've talked about, and very much like the lateral gene transfer story that we've talked about before. In this case, it is now very clearly shown that what is going on in flu is that you have these 8 chromosomes from one flu strain, and some of the time, an individual, either animal or human, can be infected by 2 different flu strains, where their genes have different histories. And during the growth in an individual cell of those 2 flu strains, you can get repackaging of these chromosomes such that the offspring flu can contain chromosomes that are mixed from the different sources. So it's called recombination within flu. It's a way of mixing the genomes of different flu lineages. And what people have done, researchers have done by looking at the evolutionary history of different flu strains compared to the severity of outbreaks of flu. So you may be aware that in 1918 there was a massive flu epidemic around the planet, killed millions of people, and then there were subsequently other flu epidemics across the years. It's about every 30 years that there's a bad one of these. 1968 was a recent one. And in every case when there's a massive epidemic of flu, it is the result of the mixing of 2 different lineages of flu to create a recombinant flu strain. And what this does is basically swamp out the immune system of humans. So the immune system of humans develops a response to a particular flu strain. And when you get these mixing between 2 different flu strains, then that particular flu strain can avoid the immune system in many individuals. And so what people do now is routinely screen, just wait one second, is routinely screen flu populations for what flavors of H1, of hemagglutinin and neuraminidase genes that they have. And this is how they predict what flu strains might be responsible for the new flu season, and then design vaccines based upon that prediction. Those designs don't always work well, but they work better and better thanks to better phylogenetic analysis. And we'll stop there.